On your video, sir? Okay, okay, sir.
So, shall I share my slides, sir?
Sir, first I will give some brief introduction about you, sir. Then after that, you can proceed with your expert lectures.
Yeah, definitely. Yeah.
Okay, thank you, sir. Sir, good afternoon, everyone. Here, welcome to the Atal Online Faculty Development Program, AI Innovations in Healthcare and Medical Technology. So today is the 5th day. Today we have one eminent personality in artificial intelligence, Dr. Srirang Ambaji Kulkarni Garu, Associate Professor, Department of Information Technology, Manipal Institute of Technology, Bangalore. Thank you very much, sir, in spite of your busy schedule, accepting our invitation and ready to give your keynote address or expert lecture in our FDP, which is aligned with your research of interest. Thank you once again, sir. I would like to give brief introduction, small introduction regarding Srirang Ambaji Kulkarni Garu. Sir completed his PhD and postdoc from University of Central Florida, Orlando, Florida, USA. Now, sir presently working as a associate professor in Manipal Institute of Technology, Bangalore. Sorry, areas of interest are wireless sensor networks, machine learning, and cyber security. Sir had a lot of publications which are indexed in Scopus, SCI, and he authored many books. In that, we have 1 year back, we have also written one book which is on brain-computer interface. We both are co-authors, co-editors, which is published by CRC Press. Sir had 2 patents, and one is granted by India, another one is USA. Sir had professional membership. He had a senior member, ACM, senior member of IEEE. So, sir had a lot of experience in teaching as well as research. Thank you very much, sir, for accepting our invitation and acting as a resource person for this FDP. Thank you very much.
Yeah. Thank you, Dr. Ravichandran, for the nice introduction.
Thanks.
Right. Yeah. I welcome all the audience for this lecture. Like sir has mentioned, right from the year 2016 or earlier, I've been working in the area of artificial intelligence. And as a part of my postdoctoral studies, I worked in the domain of healthcare. And the problem which I tried to address was prediction of COVID-19. And it was an open problem during those days. So my today's talk will also focus on COVID-19 and how the vaccines were developed and what AI models were part of it. So, and it's a very rich field, the field of genomics. So I hope it will provide a lot of insights for the young research community to further explore. So can I start my talk, sir? Yeah, thank you, sir.
Thank you.
Yeah, I'll share my slides.
Sir.
Yeah. I'll just share it. So are my slides visible, Professor?
No, sir.
Now, sir?
No, sir.
Oh.
You have not shared your screen. I think has to give
Okay.
host controls. Uh,
The window you selected is invalid. Okay, fine. I'll do it once again. Uh,
okay.
we keep on using more of Microsoft Teams, so these different setups. I'll say entire screen share.
Yes, sir. Entire screen,
Um,
sir. Yes.
now is it visible, sir?
Yes, sir.
Perfect.
Excellent. Yes,
Yeah. So I'm going into the full mode. Yeah.
yes, sir.
So the topic for today's discussion is AI for personalized medicine and genomics. So we'll be trying to look into this and how AI has helped these two fields. Like almost all of you know, like that the discipline of computer science that as we have, it has grown more because it adds efficiency to every process. Let us take manufacturing, or let us take drug discovery, or let us take genomics, or let us take space. Every field, our computer science has traditionally added efficiency. Now, this has further grown, right, when we have come into the field of artificial intelligence. Uh, I hope most of the people have a background in artificial intelligence, but if not, a brief summary like how the present AI that we have, and it's more of a machine learning and deep learning. The term AI was quite long ago, like somewhere in the year 1956 in an international conference, and they thought that computers or software will be able to mimic the human cognitive abilities. Like the greatest gift that we humans have here is we can think, we can reason, and we can take our own decisions without anyone's intervention. So they thought that the computers or artificial intelligent algorithms would match these capabilities. But somewhere around 1990s, it was realized that this thing that we are exploring is a futile approach, and that's not possible. At the same time, somewhere in around 1996, Dr. Andrew Ng, who is a associate professor at Stanford University, during those days he was working in Google's DeepMind, will be coming later again to Google DeepMind. And what he thought, like, look, the most successful kind of algorithms that we had, or which he thought was possible, was supervised learning, right? And he said, like, if I scale up this supervised learning, the training data set, if I scale it in a very huge manner, and then I query it with some data, there is very high chance or very high accuracy, it will understand that and predict the outcome. So he developed this and he called this as deep machine learning algorithms, or we call them as our scalable neural networks. So he designed this, and that really took the growth of AI to the next dimension. All the AI that we see today are nothing but the inventions or the developments or innovations that have extended from deep learning algorithms till the present thing that we are witnessing, the revolutionary chat GPT. And if we read the news, there are some unsubstantiated claims that they have even come up with artificial general intelligence. It is not yet substantiated. Few scientists from OpenAI are claiming. So we are in that era now where we are generating data or information in a manner that is conducive for the human to accept it. So we are in that area. Okay, with this background of, and we also have unsupervised learning where the accuracy levels are high, but it is still useful in other domains. There is a third pillar, which we call it as the reinforcement learning, and it also has many applications, especially in terms of our autonomous vehicles and robotics and automations. So AI is something that is going to take the humans to the next era. Now, the today's topic for discussion that I'll be taking here is uh, One second, I'll just minimize my. Yeah, I'll be talking here is about COVID-19. As sir mentioned that I did my postdoc in University of Central Florida, and I came up with an algorithm, and that algorithm was able to predict something which will happen for the next day. And I initially thought like, let me apply to predict the temperature because Florida was a place where many space launch vehicles would be launched because that there is the space launch vehicle facility. And I said temperature plays a huge role. But my supervisor said, no, you're working in a healthcare department. You should see that your algorithm works with something like healthcare related thing. And COVID was a very much prevalent thing. And after I joined in 6 months, the Delta variation came. So I thought, why not apply my algorithm for COVID prediction? And if it is able to predict the next day's cases, then we can make a resource provisioning, like how many vaccines we need, or how many medications we need, how many masks we need, how many PP kits we need. So I applied my algorithm for that domain. So when I was invited to give a talk for this, I thought it's but natural that I should speak on COVID-19. So all of you know that COVID-19 is an infectious disease and it was caused by the SARS-CoV-2 virus. And all of you know that most of us have suffered during this pandemic, directly or indirectly. And what are the symptoms that WHO has categorized this year is when people are infected with this virus, they exhibit respiratory illness and sometimes recover without special treatment. Like uh, we might have had people who got COVID or coronavirus and they just came out of it, right? But however, some of the people also fell seriously ill and required medical attention, especially in the first phase. It took the shape of pandemic and the mortality rate was very high. Also, the people with morbidity, like existing diseases like diabetes, chronic respiratory disease, or cancer, these people, the chances to develop the illness was more severe. And there was no age restriction, like it might be an old person who may acquire COVID-19, or he may be a young person. So any of these persons would get infected by this and die. Yeah. Now, this COVID-19 had an unprecedented impact on humanity, and we categorized it as a pandemic. You know, uh, excuse me, I'm hearing some background noise. If anyone has anything, please say, or please switch off your mics.
मनीष सर ऐसे करना है मेरे पास सिर्फ एक ही बच्चा आया है हाँ थर्ड सेमेस्टर का
Hello.
वही तो ना वो अब दिखाना भी पड़ेगा वो कैसे करेंगे कैसे
Hello.
नहीं
I request you all to please switch
बट
off your
अपना
mics.
नहीं मैंने रिपोर्ट एक ही बच्चे ने करा है सर ऐसे ऐसे मेरा अच्छा आपके फिफ्थ सेमेस्टर
Yeah, yeah. Now, when we look at the global morbidity and mortality, like as I said, morbidity is more of a term which we describe for illness or disease, whereas mortality is a term for death. Okay, if we see the COVID-19 impact, there were around 6 million deaths and 600 million reported cases as of 2024. And there were disproportionate effects on vulnerable populations, including the elderly and those with comorbidities. So what happened with this COVID impact was it overwhelmed the healthcare systems in almost every country, maybe developed or developing. The developing countries had a bigger impact, but even the developed countries suffered. And what did this lead to? It led to the delays in non-COVID treatment. So it was a pandemic of very disproportionate. Just a second, I'm not able to move my slide. Yeah. So what was the impact of COVID on humanity? The world went into a recession. Like World Bank reported that the global GDP of approximately $84.97 trillion in 2019 shrank by 3.5%. And that resulted in a loss of $3.94 trillion. Widespread job losses and business closures in sectors like hospitality, travel, and retail, and disruptions of supply chain and increased inflation because people could not transport the essential goods, and it all started impacting the humanity very hugely. Now, when we in the background of this, there was a great confusion, like how to develop a vaccination in such a short duration, because the earlier vaccines that were developed, right, took at least a decade. So the worldwide scientific laboratories, especially the what you can say, the laboratories who work on genomics and biomedical things, they started rushing against that time.
நீ வரவ ஏன் கான்ஃபிடென்ட் இல்லாம பேசிட்டு இருக்க? நான் போயிடுவேன். உனக்கு என்ன பிரச்சனை? நீ எங்களை கூட்டிட்டு போவேன்னா சொல்லு, நான் வெயிட் பண்றேன். ஆமா கரெக்ட். ஐயோ இல்ல கரெக்ட்.
I see a lot of people talking. Please, I would request not to talk. Yeah. So, okay. Where there is an option to mute all, I'm not aware. Like, do I have an option for that? I'll just try to check it out. Okay, fine.
ആടുന്നുണ്ടായിനല്ലോ കഴിച്ചിരുന്ന നല്ല ഉറപ്പായിന് നല്ല ഉറപ്പടാ ചെറുങ്ങനെ തറച്ചിട്ടാ അച്ചാറുള്ള ഒരു വലിയ അച്ചാറില്ല മസാല കറിണ്ടാക്കുന്ന ഒന്ന് രണ്ടെണ്ണം നാലെണ്ണം ഉണ്ടാവുള്ളൂ മസാല കറി ചിക്കൻ്റെ അതേ പരിപാടിയാണ് ഓരോന്നില്ല അത് ഇത് വെച്ചല്ലേ ചക്ക ദേശം മസാല കറി ഉണ്ടാക്കും അല്ലേ
Yeah. So in this race for vaccination, right, there were two big giants, like one was Moderna and another was Pfizer. And they thought that the mRNA vaccine was the most promising one and which would be developed very quickly and with great deal of efficacy. So what is the outcome here is like global vaccination campaigns from Pfizer, Moderna, our own Covaxin, and another vaccine from India. These saw that they reduced the severity of the illness and mortality. So I'll be speaking about Pfizer because I myself had taken a lot of Pfizer vaccinations, right, when I was in US. So what is the story like how Pfizer developed its vaccine for COVID-19? As I told at the beginning of the discussion, if you want efficacy, if you want speed, the only way to do it here is to take the help of computers and smart or intelligent algorithms. So as Pfizer scientists raced to develop their COVID-19 vaccines at a record-breaking speed, they turned to innovative artificial intelligence tools to help this machine. So Pfizer was in collaboration with BioNTech, a German biotechnology company, and that was specializing in mRNA technology. This technology was still under development, but people said, let us apply it for developing a vaccine for COVID-19. These slides get stuck. Just a second, my slides are not moving. I'll just take a. Just give me a couple of seconds. Here, yeah. I'll present it in this mode because there is some problem. Okay. So in molecular biology, that is messenger ribonucleic acid, what happens? A single stranded molecule of RNA that corresponds to a genetic sequence of a gene and is read by a ribosome in producing a or synthesizing a protein. So there is a genetic sequence, and this mRNA is a single stranded molecule that becomes a part of this genetic sequence of a gene. And what are these ribosomes? They are nothing but they are macromolecular machines found within cells that perform biological protein synthesis. As we all know that life is dependent upon the synthesis of protein. So ribosomes play an important role, and mRNA plays an important role in defining or becoming part of a genetic sequence. Now, in background of this, we should know something like a spike protein. And what is this spike protein? This spike protein is a key protein that was found on the surface of SARS-CoV-2 virus. And this is the thing that causes COVID-19. So isolation of this spike protein was very important. And it is critical for the virus to infect the human cells, and it's also the main target for. So naturally, spike protein was one of the main targets for the companies which are going to manufacture COVID-19 vaccines. And where this spike protein is located, it's located on the outer surface of the virus, and it has like a crown-like appearance. And since it has like a crown-like appearance, it got the name like COVID-19 or coronavirus. That is the crown. Now, what is the role of this infection? This spike protein enables the virus to attach to human cells by binding to a receptor called as ACE2. ACE2 stands for angiotensin converting enzyme 2, and which is found on the surface of many human cells. So this spike protein tries to attach itself to the receptor called as AC2, and this receptor is especially common in lungs, heart, and blood vessels. After binding the ACE2, the
Atlantic
spike
color,
protein undergoes a structural change that facilitates the fusion of the viral envelope within the human cell membrane. So once it has got stuck to the spike protein, once it binds to ACE2, then the viral envelope has to be relieved into the human cell membrane. This allows the virus to, what you can say, release its genetic material into the host cell, initiating the infection. Now, why we are discussing this initiating of infection here is we want to generate a synthetic mRNA, and we want to see that the antibodies are produced for this by the human immune system. So when later coronavirus infects a human, our body is ready with, what you can say, immunity cells. So that's the story behind this. So as I said, the spike protein is used as an antigen in most of the COVID-19 vaccines. And like mRNA vaccines, like both Pfizer and Moderna, they basically instruct the cells to produce a harmless version of the spike protein. The immune system recognizes the protein, like as I said, like as a foreign body, and generates antibodies against it. If the actual virus later infects the body, the immune system is primed to attack it. So by putting this, what I can say, synthetic mRNA into the body and attaching it to the receptor, we are generating greater immunity in the host. So how are these antibodies neutralized? So antibodies generated by the immune system target the spike protein to block its ability to bind to ACE2 and thereby effectively neutralizing the virus. So what is this ACE2? It stands, like as I said, to angiotensin converting enzyme 2, a protein that plays a key role in several physiological processes, and it's particularly in the regulation of blood pressure and maintaining cardiovascular health. So you see that most of the people who were affected with COVID-19, the morbidity is affected primarily to these organs like lungs. Those who were infected with lungs suffered from pneumonia and acute respiratory disease syndrome. We call it as ARDS. For heart, it was myocarditis and heart damage. For blood vessels, they had clotting issues, and even kidneys and intestines had organ dysfunction. So if you see that most of the people with COVID had morbidities affected with these organs. Now, if you think of, you can think of ACE2 as a doorknob. And as we know that doorknob usually helps us to open the door. And this doorknob was used by the SARS virus to enter the cell and cause infection, disrupting the protective functions in the process. So the ACE2 receptor played a very important role, both in terms of spike protein getting joined to it and infecting it with the virus, and so on. So now we should see how this vaccine design and optimization took place. So the RNA structure optimization, AI was used to identify and design the optimal mRNA sequence. As I said, the genetic sequence, the single strand is a mRNA, and AI was used to generate a effective mRNA sequence. And this involved predicting how different mRNA sequence would behave in terms of stability and translation efficiency. Because this is something like a synthetic mRNA and this has to be injected into the human body. So its stability is important, and also how it can be transported and fed into the cell. So translation efficiency was also very important. The mRNA in the vaccine provides instructions to the cells to produce the spike protein of the SARS-CoV-2 virus. So once these spike proteins are generated, that means the immune system will recognize these spike proteins, and then they start producing the immunity cells. And once that has been done, then the body will be prepared if there is a what you can say infection due to SARS-CoV-2 virus. As I said, for this to work effectively, the mRNA that has been synthesized should be stable, that it should remain intact until it reaches the cell. It should be translatable, that is, enable in efficient production of the spike proteins in the cells, because once spike proteins are generated, the cells will detect that this is something we have to defend, and they start developing the immunity, what you can say, cells. And also this mRNA should not result into side effects, like it should not result into allergy or alternative immunity responses from the human body. So there was lot of AI involved in generating this mRNA. And both these companies, they took help of the most cutting edge technologies that were around during those days. So the first thing that they relied on was, like as I said, Google AI's DeepMind tools. DeepMind has been in the forefront of developing deep learning technologies, and the famous one is called as AlphaFold. Do you know that the 2024 Chemistry Nobel Prize was awarded to John Jumper, Demis Hassabis, and David Becker? And this all goes into their work in developing or showing the interactions of protein structures in a 3D format. Okay. And basically, this work was carried out at Google DeepMind in London and at the University of Washington in Seattle. So what does this tool, which we call it as AlphaFold, the present version is AlphaFold 3. What it does is it has revealed millions of intricate 3D protein structures and is helping scientists to understand how molecules interact. As I said, protein synthesis in turn leads to cells, and cells, how they interact is modeled in a 3D fashion by this AlphaFold. So as I said, proteins underpin every biological process in every living things. So proteins is the basis for life, and it is made up of long chain of amino acids, and each such has a complex 3D structure. But figuring just one of these complex structures can take several years, and it may cost of hundreds of thousands of dollars. Can you just imagine? Just this protein structure in terms of amino acids and studying its 3D structure will cost millions of dollars and will take years to analyze it. And this problem was solved by AlphaFold in 2020, and they were able to predict protein structures in minutes. Earlier it was years, now they were just in minutes, and they also predicted it with remarkable degree of accuracy. This was something like marvelous, and this was happening at the same time when COVID had launched its attack. Like COVID was spreading or was going to spread fast. So this helped researchers understand what individual proteins are and how they interact with other molecules. So, so what happened here is, since we could visualize these protein interactions in 3D structures, it saved a lot of valuable time and resources, and it also redirected advancing researchers to solve more other issues related with COVID-19. So how does this AlphaFold work? What is the AI behind this AlphaFold? As I said, AlphaFold was developed in DeepMind, and DeepMind has been very famous for developing deep learning techniques. And as you know, one of the important techniques are transformers and attention mechanisms, right? So this chat GPT that we see today is also based on the transformer technology. Later we'll see whether it's part of an encoder or a decoder uh, transformer technique. So what happened here is, by leveraging evolutionary data and geometric modeling, so this AlphaFold predicts protein structures with unprecedented accuracy, and it is transforming, like as I said, fields of drug discovery and molecular biology. So what are the inputs to this AlphaFold network? It consists of multiple sequence alignments, which we call as MSS, and pairwise features. The amino acid sequence of the protein is the fundamental information that dictates how a protein will fold. So you have a huge amino acid sequence, and that provides fundamental information like how protein will fold. And what is the output of this AlphaFold? The output represents a predicted protein structure, and the confidence of the structure matches where the protein structures are mostly coordinates of proteins. So then these 3D structures that have been generated of these proteins, these can be visually rendered. So as I said, the present version of AlphaFold is 3, and once these guys from DeepMind won the Nobel Prize. AlphaFold 3 and its existing source code and data sets have been made as a open source. So all the researchers in this forum can just go to the sites and download it, and they can use it for their research. But how did this AlphaFold 1 and AlphaFold 2 develop? The AlphaFold 1, it basically used the earlier forms of deep learning neural networks, which we call it as the convolution neural networks. And it was good in learning local parameters in data, like recognizing small local features in images. In case of a protein folding AlphaFold 1, it used CNNs to process protein sequence and predict their structure. But however, with AlphaFold 2, it used transformers, a type of neural network structure designed to capture long range dependencies in data.
मुझे लग रहा था वो सर आएंगे
ऐसे क्यों लग रहा था?
क्योंकि 2:30 बजे तक मैंने कॉपी का वेट करा
ओके
मतलब बट ये कि साथ
वो तो फर्क रहता ही।
जब कोई मिला ही नहीं, कोई आया नहीं, तो फिर मैं आ गई कॉपी इसलिए तो
I would request everyone to mute their microphones, please. Amisha Raj, please mute your microphone. Yeah.
का काम अच्छा रहता।
Yeah. As I said, AlphaFold 2 was using transformers, and transformers are a type of neural network architectures that are designed to capture long range dependencies in data. And as I said, this is especially important in protein structures because the relationship between distant amino acids play a big role in determining the overall protein. Yeah, apart from this, the transformer architecture that was used in AlphaFold was called as the evoformer block. So what is this evoformer block? This is the key component of AlphaFold 2, and as I said, it's based on the transformer architecture. So what is the transformer architecture? It is basically a deep learning model for handling sequential data. So till now, what were the best deep learning models to handle sequential data? They were type of recurrent neural networks like LSTM, which we call it as long short-term memory, and BiLSTM, bidirectional long short-term memory. These were the most popular ones to handle sequential data, but they had limitations, right? And what limitations means? They could handle limited amount of what you can say instances within the sequential data, but these transformers in turn used a mechanism called as self-attention, and these could capture relationship between all elements in a sequence at a once, some kind of parallel mechanism. So transformers really revolutionized the thing. So why transformers were so popular? Like as I said, parallelization, instead of looking token wise, they could process all the elements in a parallel manner. They help in long range dependencies, they could capture relationship between distant elements in a sequence. And this was one of the major advantages over RNN, because RNN, when it tried to capture long range dependencies, it suffered from something called as the vanishing gradient problem. So what is this vanishing gradient problem? It's a common type encountered in deep neural networks, especially RNN and LSTM. And it occurs when gradients, usually when we see them as weights of our neural network, they start becoming exceedingly small as they are backpropagated through the network. And this results in slow or ineffective learning, because the weights that are getting modified have very low range of weights or very less weights. And this in turn reduces the learning rate of the network. So this was overcome in transformers. The next advantage was flexibility. Transformers have been used in a wide variety of tasks, like they have been applied from NLP to protein structure prediction, like what we are studying right now, AlphaFold 2, and even in image recognition. So as I said, this transformer in NLP, we have two major applications. One was called as the BERT, which stood for bidirectional encoder representation from transformers. And it learns bidirectional context by masking certain words and predict them based on the surrounding context. And the next one is GPT, or the chat GPT, which we are more familiar with. It uses the decoder part of the autoregressive text generation, where model predicts the next word in a sequence based on the previous words. So that's why we call it as a generative model. It is able to generate the next word based on the previous words. So what is this self-attention? This self-attention in the Evo former, that is the transformer model of AlphaFold, it captured long-range interactions in the amino acid sequence, understanding how different parts of the protein affect each other. This information is then used to update. We have something called as pairwise distance matrix, which gives the spatial information. And finally, the triangle updates to see that they are geometrically valid and they don't violate the principles of triangle. Now, when these combined processes, AlphaFold was further used to predict protein structures with high degree of accuracy. So right now we are studying it for disease understanding, but this AlphaFold has numerous applications, like it helps in drug discovery, it helps in biological research, and it helps in disease understanding, and it also helps in enzyme engineering. So AlphaFold was one of the major tools applied by Pfizer and Moderna for the generation of the COVID-19. The next thing that you see is we have something called, as you might have read some articles in MIT magazine and all, we have something called as a genetic Caesar, by where you can cut the genetic sequence, modify it, and rearrange it. So these type of tools also went into the design of the COVID-19 vaccine. What was it? It was CRISPR-Cas9 guide RNA design. And this was basically a revolutionary tool in genetic engineering. It allowed us to edit the DNA structure by targeting specific sequence in the genome. So what were the two main components of this? One was the Cas9 enzyme, a protein that acts like molecular scissor that was cutting DNA at specific location. And we had a guide RNA, or which we call it as a gRNA, a short RNA sequence that direct the Cas enzyme to correct the part of the DNA for editing. So these were the two major roles. So what was the role of this guide RNA or gRNA? So this gRNA was designed to be complementary to the target DNA sequence. So when gRNA binds to a target, it guides the Cas9 enzyme to that region where Cas9 then cuts the DNA. This allows for targeted DNA editing. This is a very superior technology, and it's going to be the kind of future of humanity. So the effectiveness of this CRISPR-Cas9 depends on how well the gRNA binds to the target DNA and how it accurately the Cas9 enzyme makes the cut. So it is based upon those. So as we discussed, guide RNA or gRNA design was critical for optimizing the CRISPR-Cas9 system for gene. And it's also the important goal of these design algorithms is how they go and attach to the specific target. Okay. And these algorithms, like as I said, help us to realize sequence composition, secondary structure, and off-target predictions to optimize gRNA sequences for precise genetic modifications. So first we had AlphaFold that help us to analyze the protein structure in 3D format. Then we had this CRISPR-Cas9, which worked like a genetic Caesar, helped us to modify the genetic sequence. And then the most important thing that a lot of neural network, what is the researchers work on, is the gradient descent optimization to refine sequence parameters. So what is this gradient descent optimization? It's widely used algorithm in machine learning and optimization for refining the parameters of a model to minimize a loss or a error function. So in context of refining, let us take our own example such as CRISPR-Cas9 guide, right? Gradient descent can be used to iteratively improve the parameters such as what you can say optimization criteria like minimizing off-target effects, improving specificity, or enhancing efficiency. The main thing in this CRISPR-Cas9 is it should go and attach to the target properly and effectively. So what is this gradient descent? Its goal is to either minimize or maximize based on the objective function. In simpler terms, you can visualize this as it adjusts the parameters by looking at the slope, which we call it as the gradient of the function, right? So if you want further simpler terms, you can visualize this like you're hiking down the mountain and your goal is to reach the lowest point in a valley. So what is this mountain? This mountain is a loss function. Now, why this mountain has appeared to us? Because of how wrong your model is predicting, because that the model has come. Now, what is this slope? The slope of the mountain is your current position. That is means what is the error that has come out of outcome of your model's learning rate? That is the slope. That is the position where you are, right? That is, you want to go down the hill, but you have to move in the opposite direction of the gradient, that is back propagation. Then you're taking small steps. So you don't want to take a giant leap, you have to take small steps, that is the learning rate. How do you modify the learning rate? So you make the learning rate more more what you can say precise or small, so that the overall what you can say accuracy of the model improves. And this process you go on making till you reach the lowest point where the slope is flat and the gradient is almost reached zero. At this point, you have minimized the loss. So this is the third thing. So for any AI model to work accurately, you have to work a lot on its gradient descent and thereby modify its parameters. So how this gradient descent was applied in CRISPR for RNA design? So as I said, off-target binding. A penalty is added if the guide RNA binds to region of the genome other than the target. If the genome, that enzyme goes and sits on the not on the exact target, then there is a penalty to it. However, if it goes to the exact cell where it has to get binded, then there is a target efficiency or there is a positive reward. Apart from this, the area where it binds is or the way these things have this envelope has to be injected into the body. The next important technological innovation was lipid nanoparticles. So as I said, these lipid nanoparticles are critical delivery vehicles used in mRNA vaccines, and their major role is to deliver them to the destination. So if you have to optimize these LNPs, right, for the design effects, the first one is that you have to look into the efficiency, like how well the mRNA is delivered to the target cell. And the next one is safety. There should be, see that it should not result into some allergic reactions or it moves to some other cells than what it was targeted for. Now, what is the role of AI in improving this LNP lipid nanoparticles design? So one is encapsulation efficiency. So the AI models were trained on data sets of LNP formulations to predict which combinations of lipids would most effectively encapsulate mRNA. because this mRNA cannot be directly injected into the body. It should be part of some fatty or lipids, and then it can, as a part of envelope, and then it can be injected into the body. The next one is cellular uptake, right? Uh, like the predictions were made to optimize the ionizable lipid composition, which is crucial for endosomal escape. What is this endosomal escape? Is once it has traveled to the target cell, how do you release the mRNA from the vesicle inside the cells? So how does it actually travel inside the cells? And finally, the biggest next challenge is stability and circulation. So AI identified lipid combinations that reduce the degradations of LNPs in the bloodstream, and PEG lipids were optimized to balance the immune evasion with biodegradability to minimize potential immune reactions, so that it could be safe to human body. So now we had nanolipid particles, which would take these enveloped mRNA, synthetic mRNA to the body. Now, among these, there were two variations of the COVID-19 which were the best, right? So we had Pfizer and we had Moderna, right? So both worked very well in reducing the mortality, but in some instances, Moderna outperformed Pfizer. Why is it so? We'll discuss at the end. So, as we know that both the vaccines used mRNA technology
Then synonyms will be there, no?
to
Like that
deliver
only flag
genetic
can be
instructions
revealed,
for
ma'am.
cells
For
to produce the spike
using
protein of the
AI,
virus, and
still
that triggered an
writer,
immune
then
response.
ChatGPT human. ChatGPT also. Yeah. But skeleton.
So, what was the initial effectiveness? Both these vaccines demonstrated high efficiency around 90 to 95%. And but however, the what you can say, the immunity of these started deteriorating. So, booster doses were required. So, which were the best, the performance across difference? First, we had the alpha and the beta variations. So, these were quite good. Then we had the delta variant, and in the delta variant, Moderna showed slightly better durability because the initial dosage of Moderna was high in terms of those weights as compared to Pfizer. And in terms of Omicron, both, that is the final versions after Delta, we had Omicron. Again, these both showed better to those coronavirus variants. So what were the key differences between them? As I said, Pfizer had an initial dose of 30 micrograms and Moderna had 100 micrograms. So the higher dose might have resulted in terms of long lasting immune response, right? The time between the next booster doses for Pfizer, it was 3 weeks. For Moderna, it was 4 weeks. So as we all know that Pfizer required ultra cold storage of less than 70 degrees Celsius, but Moderna was possible with minus 20 degrees Celsius. So both of them worked very well, but Moderna had slightly a little big advantage. So this was about this. So this is the summary of what all we discussed, that the major takeaway from this was mRNA was the, what you can say, the technological innovation. How did this AI help here is AI helped in predicting the stability and other things of these mRNA sequence, and it also helped in predicting the preclinical testing, clinical trials, and what were the variant responses, because we have to come up with generative data to see how these new mutated variations of these viruses get infected by this. What were the challenges in terms of manufacturing, because these viruses, these vaccines were not produced in terms of limited thing, but they were scaled for huge number of vaccines. So what were the challenges for them? How, when you scale, AI models were used to predict the challenges for scalability. And finally, we had the post-market surveillance, like what was the efficacy of these viruses, what were the mortality rates, were there any side effects. So in all this, the AI technology helped. And as we discussed, the Google's AlphaFold was one of the key players, along with genetic Caesars like Casper. Now, coming to the next topic, that is AI for personalized medicines and genomics. Uh, if you look into the future, like most of the medications that we produce for a particular section of population may not be relevant for the other section of population, because it's a type of bias that gets introduced into the artificial intelligence models. So most of these vaccines may be true for the white population, but they may have some other impact on the Asian population. And if you go into further depth, there are diseases which affect a person individually. So in this case, what are the roles of AI and how medicines for a particular human populations or individual humans can be developed? So the example that we consider here is non-small cell lung cancer, or it's called as NSCLC. So as we know that lung cancer is one of the leading causes of cancer. It may be due to pollutions, it may be due to smoking, or it may be due to passive smoking, and it accounts for more than 85% of cases. So what are the things that affect these cancers? Like, as we know, cancer cells are generated within the human body. There is a cell manufacturing factory and it gets disrupted, and once it gets disrupted, it starts producing abnormal cells, and these abnormal cells or mutated cells take the role of cancer, and then this travel through the lymph nodes, and then they start destroying the host itself, that is the human body. But in case of this NSCLC, the major mutations occur because of EGFR, ALK, and KRAS. So what are these? So EGFR, it stands for epidermal growth factor receptor, and it promotes tumor growth because based on angiogenesis, that is a new blood vessel formation, and resistance to apoptosis, that is cell death. So it is basically promoting tumor growth. The next one we have is ALK, that is anaplastic lymphoma kinase. This basically rearranges or enhances tumor cell proliferation, invasion, and survival. The most dangerous is KRAS, that stands for custom rat sarcoma viral oncogene. And what it basically does is it drives aggressive tumor growth and is often associated with poor prognosis. That is, its growth is not so much easily identifiable. And the tumors with this KRAS mutations are very historically difficult to treat, as KRAS was considered as undruggable for many years. So now how AI is used in generating personalized medicines. So first we need to have genomic analysis. So we try to see the EGFR mutations, and we try to analyze how they can be targeted by specific drugs. The tools that were employed here was the IBM Watson. So IBM Watson, the AI tool from IBM company, has multiple packages for artificial intelligence and deep learning. They also have what you can say curated data sets. So using IBM Watson AI applications, you can generate AI models whose accuracy is better, and 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 it's also trained on curated data sets. So the chances of errors are minimized. This is very much true in case of healthcare, because if the model predicts something which is wrong, it may affect the human, what you can say, the human participant. Finally, we had predictive modeling. As I said, AI predicted individual responses to immunotherapy drugs like Keytruda based on PD-L1 expression levels and other biomarkers. So how was the treating optimization done? It was done using reinforcement learning models. What are these reinforcement learning models here is, as I said, it's the third pillar of artificial intelligence, and it's based upon what we can say is called as learning with the critic. That is, there is an agent in a simulated environment, and if the agent takes a right decision, it is rewarded. If it takes a wrong decision, it is punished. And every event leads to the agent moving to the next state. So if the agent has to reach to the terminal state, it is a series of success that it has got, the rewards that it has got. If it is punished frequently and it's not able to navigate to the terminal state, it will remain in some indeterminate state and it cannot reach the terminal state. So these were used in the optimizations of the treatment. And finally, we have monitoring and adjustments, like the dosages have to be adjusted based on the patient's responses using variable devices and medical records. So all these things were applied in the personalized medicine, this for lung disease. So what was the outcome when we had these personalized therapy? The first thing here is the survival rates improved drastically, right? Patients received AI guided treatments. They had better progression free survival rates as those compared with standard treatment plans. As you know that most of the hospitals have standard treatment plans, and any patient who falls in that range is given that treatment. But personalized treatment increase the survival rate. Then it also had faster diagnosis because actionable mutations and targeted therapies were better analyzed with AI. And finally, we had personalized therapy that included specific genetic and disease profiles, thereby improving efficacy and reducing the side effects. So the final LNP, as we discussed in our previous section, that was used in this was mRNA and reduced immune overactivation and stability in cold storage. So these were some of the things that we saw when we studied mRNA. And as I said, all these vaccines that are produced, they have to undergo a lot of data analysis, and also the delivery systems have to be taken into account. So this was my discussion about how COVID-19 produced a pandemic challenge and how the vaccines were produced quickly and AI played a critical role. Now, so AI played a critical role and it helped to produce these vaccines in a very short duration, helped in preclinical trials, helped in clinical trials, helped in scalability, and also helped to take the survey or feedback from the market. So AI played a very critical role in pandemics like COVID. Now, why did we take the example of COVID? Because a pandemic is a problem which affects globally and it's uncontrollable. So AI played a critical role, and big thanks to Google's DeepMind AlphaFold, it helped to analyze the protein structures in a very effective manner. So with this, I conclude the talk. If there are any questions, I'm open to it. Any questions related to this, I would be happy to answer.
Good afternoon, sir.
Yeah.
Good afternoon, sir. Actually, what I have this topic which I have selected for my PhD was smart drug detection for diabetes. So basically, I want to design a personalized medicine suggestion tool. So, what kind of algorithm would you suggest, sir, for designing this kind of tool? Because these days there are n number of medicines which are available in the medical field for diabetes.
True.
But few patients, for them the medicines are not suiting. And what we have seen, doctors are suggesting the medicines without knowing the suitability to the patient. So after using the medicine for 15 days or a month, patients are having many side effects of that medicine. And due to this, the health is affecting a lot. So I want to design a personalized medicine for each patient. So can you please throw some guidance on this, sir? How should I proceed for this?
Yeah. When you mean personalized medicine, you want to even change the chemical composition of that medicine?
No, sir. Whatever medicines are there, however, they are good enough. But according to the body type, according
Yeah.
to your daily routine or your habits, eating habits, whatever dietary food you are taking.
True.
Based on
So
that.
yeah, basically, see, for diabetes, you can, if you're a doctor or if you have access to a data set, I would recommend that you generate a sample data set. Maybe let's assume that you have access to some 100, what you can say, records of your patients. Let us hypothetically assume, and for these people, you want to give medication. Like if I understand you clearly, for diabetes, you want to increase or decrease the potency of the medicine based on the patient's information, right?
Yes, sir.
So see, now for all these algorithms that I explained, the deep learning algorithms, you need huge amount of data. Data that's of the order of, even if you have a 10 lakh records data set, that would be great. Okay. But what did I take in my example? Let's assume you have just 100 records. So what you can go here is you can go for something called as, are you aware of AI models and all, madam?
Yes, sir.
You can go for generative adversarial networks, and you can generate your own synthetic data set.
Sir, can you just repeat it? Generative?
Generative adversarial networks. If you want, I'll type it in the chat. It's called
Yes,
as
sir.
GAN, or it's called as generative adversarial network. Using this, you can generate your own synthetic data set of, let us assume, from 100, you can spike it up to a lakh or 2 lakh. Okay. And then I would treat it like a multi-class classification problem, or you can, multi-class classification problem. And then I would tell you to apply deep learning neural networks.
Yes, sir.
And once based on that, you can say for these all parameters, this is the medication that can be classified. Like you can categorize, like if I would say some medication with 50 mg, some medication with 100 mg, some medication with 150 mg, some medication with 200 mg, I would classify it as 0, 1, 2, 3. Okay. And then based on this classification, I would say if these are the parameters and this threshold has been breached, then this medication has to be issued to it.
Okay.
So you can generate your own model, and then based on those model, feeding the, like a new patient comes and he has some parameters as measured by the medical records, you can fire this query to your model, and your model will give a specific medication.
Okay. So
And being a domain expert, you already know that this is good or bad, and then you can take a call. But as I said, personalized medication, including the chemical potency of the medications, would be far more effective. Like giving the exact delivered dose would be more effective, but that's pretty complex. We should go with medications available in the market. I hope I've answered.
Actually, I am from the field of engineering, sir. So,
oh, fantastic.
So that's why it will be very difficult for me to go with the chemical formulas.
Yeah.
So do I need to take the record histories or records of same patient for the trial? The past records. And
okay. Yeah, see how it happens here is due to the prognosis. If his disease has increased or decreased, then his record in the data set will change.
Okay.
Okay. So initially, I don't think so. It will change very drastically. Diabetes, it will take some time. So you can keep those data sets as it is. But if you're from an engineering background, I would say that your findings you share with a domain expert, a doctor, and seek their advice. Like what you're doing is makes sense. Okay. Yeah. Thank you. Anybody else? Any questions?
Thank you so much, sir.
Yeah. Thank you.
Yes. Dear participants, any other questions? Any questions?
Sir, can I ask one more question?
Yeah, please go ahead.
Yes, ma'am. Welcome.
Sir, from where can I get a good study material for this kind of research? Which site would you prefer, sir?
See, as I said, if you can contact some clinic and they provide you some limited data set, that data set, if they provide in Excel format, it's good. If they don't have it in Excel format, in some written format, you can generate your own data set. And also you can see that some of these, what you can say, records that are provided by clinics have information in variable format, in general text. Okay, so you have some models which will convert them to numeric values, right? So if you want to begin with, we have that machine learning tool called as SkyKit. You know about it? SkyKit.
No,
Killer?
no, sir.
Yeah.
Can you just type it, sir?
Yeah, I am just typing it in the message box. You can use SkyKit, and
Oh,
it has already
Scikit.
some
Yes, sir.
predefined data sets. Just for your initial experimentation, you can use. But I would say if you get some live data set, because genetically the Indian and Mexican population are more prone to diabetes.
Yes, sir.
So, and these data sets which I say are available in libraries, they are mostly from the European or American population. So I would recommend you to contact some clinic and try to generate your own data set, because generating your own data and what you can say, curating it is also a kind of research.
Yes, sir.
And there are
Thank you.
journals which publish only the data that you have generated, including journals like Elsewhere and all. I think the journal name is Data or something. I don't remember, but I can share it later. Yeah.
Okay. Thank you, sir.
Thank you.
Any questions? Participants? Okay. Now it's the time for concluding our session. I would like to thank Sriram Ambaji Kulakarni Garu, Associate Professor, Department of Computer Science and Engineering, Manipal Institute of Technology, Bangalore, for his wonderful session and enlightening our participants with his excellent delivery of lecture. Thank you very much, sir.
Thank you, sir.
Thank you, sir. Dear participants, have a nice day. At 4 p.m., you have another session. Thank you very much.
Yeah, thank you.
Right.